Hello everyone. Today I'm going to talk about my project, High-Performance Evolutionary Algorithms for Online Neuronal Control. This work is done in Harvard Medical School and Washington University in St. Louis. Our lab is a visual neuroscience lab, and we are trying to understand the meanings of the neurons in the brain. Our field starts from the 50 years ago Nobel Prize study of Huber and Binxu. What you're looking at is a shining bright bar in front of the eyes of a cat. And what you're hearing is the spike output from a neuron they're recording. You're trying to find what configuration of the bar can elicit the highest response or the most frequent sound you are hearing. And this is the quest of the optimal stimuli. The search continues after 50 years, and our lab is also following this paradigm. We think the optimal stimuli represents the contents of the neuron. Unlike Huber and Binxu, we are studying higher visual cortex in monkeys, like V2, V4, and posterior IT. These areas are known to respond to images like textures, shapes, and objects. So to find the optimal stimuli for these neurons, we need a more comprehensive image space. So we are using a generative image model, such as GAN, to parameterize the images. Moreover, we need a more efficient search method than manually choosing them. And this is the topic of today's talk. In previous works, we have proposed a method to search for optimal stimuli in the deep image generator. Briefly, we choose a bunch of latent codes and map them to images using a generator, and then show these images to the monkey, and then record the neuronal response using electrodes implanted in visual cortex of the monkey's brain. Then we count the spikes and use the spike count or firing rate as the scores for each latent codes. And then the scores and latent code are sent into the optimizers to propose the next batch of latent codes. So a little bit of formulations. The image space we're working at is parameterized by this generator, which maps latent code Z to image I. And the 4,000 dimension latent space is our search space. And our objective is to optimize the neuronal response in the monkey. We think of this response as a noisy function of the image. We denote F as a scalar function of the image, the mean response, and epsilon is a fluctuation. As we know, neuronal response are really different, could be different from trial to trial. And our problem is to find the maximum of function F over the manifold parameterized by the generator G. What are our challenges? Firstly, the search space has a high dimensionality. We have 4,000 dimension search space. In contrast, we have a really limited function evaluation time. We have only 1,000 to 3,000 number of function evaluations per session. Finally, we have huge noise in function values. The standard deviation of the noise could be as high as a signal sometimes. And this unique technical set challenges force us to choose wisely what optimizers we use for this problem. In previous works, people have used genetic algorithms or gradient descent to solve this activation maximization problem. And here we want to benchmark and survey a wider range of evolutionary algorithms and see which one works the best. We're going to first do in silico benchmarking using CNNs as models of the ventral streams of the neuron, of the brain. And we are going to use units from the CNNs as a model of neurons recorded from the brain, and then optimize the optimizers and see which one works the best in silico, and then deploy that in vivo to validate if it's really outperformed the previous baselines. As a setup, we use 2 network models and choose 7 layers per model and 25 units per layer. And we use these 3 different noise levels. We add the Gaussian noise to the output of the unit. And there are 0 noise, 0.2, and 0.5 noise. And here are the effects of noise on the signal. We choose 12 different optimizers. And they're all implemented in the Narrowgrad by Facebook. We use the default parameters for each of these methods. We find that CMA and diagonal CMA excelled in this competition. They have reached really high activations compared to all the other candidates. And this inspires us to see if other variants of CMA-ES algorithms could also work well. We choose the Cholesky CMA-ES and compare that to CMA and diagonal CMA. And we also compare them to our previous baseline, genetic algorithms. And we find that all types of CMA algorithms outperform genetic algorithms. And it increased the max activation by 96% in the noise-free condition. And the performance gap is larger for deeper units, which are known to be harder for optimizers to work. Also, the performance gap is larger for lower noise condition. And in the high noise condition, the CMA type algorithms increase the max activation by 46%. Next step, we deployed CMA-ES, specifically the Cholesky CMA and genetic algorithms in vivo and see which one works better. And we find indeed, Cholesky CMA-ES works much better than genetic algorithm baseline. And it increased the max activation by 44%, comparable to the 46% in high noise condition in silico. And here is one example evolutions we recorded in vivo. And we have already used CMA-based evolution algorithms to publish a few papers. And you can see the rapid increase of fire rate throughout this evolution and the clear image visualizing the preferred features of the neuron. So why CMA algorithm works so well? One hypothesis is the namesake, covariance matrix adaptation. So to validate that, we measure the condition number, kappa, of the covariance matrix C throughout the evolution. What we find is that for Cholesky CMA-ES, original and diagonal CMA-ES, even at the end of evolution, the condition number is really close to 1, which means the covariance matrix doesn't really change, and there's no anisotropy in the search dimensions or the exploration directions. And we notice that diagonal CMA has a larger condition number, which means it has slightly larger anisotropy in which direction explored. But even so, it performs worse for deeper units. And which means for this such high dimensions, a more effective covariance matrix adaptation may not be better. Next, we want to know what does the evolution trajectories look like. We perform the PCA onto the trajectories and see what are the projected trajectories looks like. Surprisingly, we find an intriguing sinusoidal structure. Each of the PC projections is a sinusoidal curve, and each one of them have different frequencies. And this is true both in vivo and in silico. So this inspires us to think of why we see this sinusoidal structure. We dig in the literature and find that high-dimensional random walks have a known sinusoidal PC structure. If you have the random walk like this and project the sequence of these onto the PC space, you will see what we see, the sinusoidal projected curves. To validate this idea, we measure the explained variance of each PCs in the trajectories we have and compare it to the series proposed in this paper. And we find a fantastic match, especially when we limit the number of steps we take to the actual generations we have. So this shows that evolution trajectories has the signature of guided random walk. And this has implications later. Next, we tested whether the evolution trajectories share a certain common subspace. We recall that the generator image space is anisotropic, which means some directions lead to a larger image change, while some other directions lead to no or very subtle image change. This is detailed in our previous ICLR paper. And the higher eigendimensions, like the blue trees, will induce a faster image change, while the lower eigendimension, like the pink trees, will induce more subtle image change. So under this framework, we projected all the evolution trajectory we have onto the eigenframe of the generator. We find that on average, the evolution travel further along the top eigendimensions than lower eigendimensions. This is also true both in vivo and in silico. This shows that the geometry of the space leaves certain signature onto the evolution trajectories. Finally, we notice another fact of the image space, which is the norm of the latent vector majorly controls the image contrast. And this is shown here. If you scale up the latent vector, you majorly change the contrast. And also we notice that if you sample linearly in the latent space, you are not necessarily sample linearly perceptually. And angular distance is a better proxy for perceptual distance. So if you sample equidistant angularly, your perceptual distance are more uniform. These two facts point to an intriguing chemistry. So we know in random walks, the vector norm will grow with evolution. And we know that angular distance is more related to our perceptual distance. So this means that throughout evolution, the perceptual step size will decay. What does that mean? Which means in each generation, we are explored at a certain distance. And that distance amounts to a larger angular distance at a smaller base vector norm. And at a larger vector norm, which is usually the case at later generations, the same exploration range amounts to a smaller perceptual distance and angular distance. This is validated by directly measuring the Euclidean standard deviation among the latent vectors in angular distance and LPIPS, perceptual distance among images. We find that both the image and angular distance keep decreasing throughout the evolution, just as we predicted in the last slide. So we learned these 4 lessons. Firstly, coherence matrix is not adapting, and isotropic Gaussian could work. And also search trajectory exhibits signatures of random walk. And anisotropy is reflected in search trajectories. And finally, the perceptual step size decay is key. So can we use these lessons to improve CMA-ES algorithms? The design principle we want to leverage is first, we want to operate on the sphere instead of the whole latent space because a higher contrast is usually good. Also, we think we can just use isotropic exploration instead of using a varying coherence matrix. Finally, we think controlling the angular distance or angular step size is key during the evolution. Since we are working on a sphere shell, there's no intrinsic mechanisms to decay the step size. And here, we propose to manually decay it and simulating the perceptual steps of decay. The algorithm works as following. So firstly, given a center, we can find the tangent space at that center and the sample tangent vectors as our exploration directions. Then these directions are mapped to the sphere using the exponential maps. And here, we keep the arc length or angle as mu t, which is the step size we are controlling. And we evaluate these latent codes and by showing them to the monkey. And we use the scores to do weighted average of these latent codes and then normalize the weighted mean to the shell as this dot. And we extrapolate using a geodesics to our next center. And this process will iterate and until we reach a high activation image. We empirically validate this method and find that it performs remarkably well compared to all the CMAES variants. And in a noise-free condition, it works the best among all the layers. In the high noise condition, it works comparably or slightly worse than CMAES variants. So in summary, our work demonstrates the efficiency of CMA optimizers both in vivo and in silico, and it has already been put into action in our lab. Moreover, we dissect the key principles underlying CMA. So the covariance matrix adaptation may not be necessary in such high dimension, but we need to control the perceptual step size decay properly and wisely. And another observation is the search trajectory could reflect aspects of the search space geometry, and which is maybe useful to study the geometry of the space. And finally, we developed Sphere-CMA, which performs better and corroborates our point that it's important to leverage geometry when developing optimizers. And this is our talk, and thanks for attention.